Today I'm here to discuss about the leftover embryo transfer technique. Yesterday we discussed about in embryo transfer technique. We discussed about the techniques which we use in embryo transfer, like um, the reproductive anatomy and what is embryo and what is embryo transfer, in what way we use the embryo transfer technique, and how to remove the embryo from a female of superior female, and placement of embryo into the reproductive tract of the female of average genetics, and what is the speciality of selecting the embryo transfer technique, like the goal of embryo transfer. Here, the goal of embryo transfer is to obtain the maximum number of genetically superior embryos in a minimum amount of time. As like this, yesterday we discussed about the benefits of embryo transfer, and we also discussed the necessary equipments for the embryo transfer, and we discussed about the necessity of the embryo, equipments of embryo transfer, and the process of embryo transfer, and how to select the donor cows, and how to select the cows having the superior characteristics, and bull selection, and the characteristics of the recipient cows, and the recipient and synchronization of the estrus cycle, and also we discussed about the activation of the hormones in the female and male, and also we discussed the breeding the donor cow, and how to flush, and the techniques used to flush the embryos, and also we discussed about the removing the embryos from the female, mm, and collect how to collect the embryos, and what is the use of penicillin after injecting the embryos into the donor, and what donor, and apart that we also have the embryo statistics. An average of 7 to 10 embryos is collected from each flush. However, the number of embryos obtained from a single flush may range anywhere from 0 to 60. Uh, how to separate the embryos? Here you can see the laboratory technique. In embryos, in laboratory, the embryos are separated from the flush media and examined under a microscope to determine their quality and the stage of development. First, we have to check whether the embryos are ready for transferring, or the healthy embryos are healthy and in active stage, so that with the help of microscope we can examine the embryos to determine their quality and stage of development. And embryo size is also plays the major role here, so that we have to examine and we have to check the size of embryo up to 0.2 mm. uh, for the embryo transfer, the only undamaged embryos at proper maturity should be transferred. And coming to the, just now we discussed embryo quality. Even yesterday also I have shown this one. And with the help of corpus luteum, um, uh, the embryo is then inserted either the left or right uterine horn, depending upon which ovary has a corpus luteum. And the corpus luteum is a structure on the ovary that secretes the hormone progesterone, which is need to maintain the pregnancy. So that first we have to check the um, uh, um, with the help of rod, with the help of a transfer rod is inserted through the vagina and cervix and into the uterine horn that has the corpus luteum. The recipient cow's ovaries are palpated before the transfer to determine which ovary has the uh, this one corpus luteum. And transfer immediately or freeze. Embryo should be transferred as soon as possible after the flush happen within the 8 hours to minimum at least 8 hours. Um, and then embryos can also be frozen for later implantation with the help of liquid nitrogen tanks, which is stored in the liquid nitrogen tanks. When coming to the summary, embryo transfer technique can be a costly one, so that the return on the investment should always be greater than the input cost. Embryo transfer should only be performed using the donor cows of superior genetics. A bull selection should be based on superior genetic as well. And if the required conditions are met, embryo transfer can be a beneficial way to produce superior cattle. Our motto is to produce the superior cattle. Not only the cattle, but also in case of human beings, same techniques will be used. This is about embryo transfer techniques in cattle. Our next topic is here, our next topic is agricultural biotechnology. We have our next topic, agricultural biotechnology, as our next topic, and also we have aquatic biotechnology. And first, today, now we will discuss about the agriculture biotechnology. What is here? What is biotechnology means? Biotechnology is the use of biological systems such as microorganisms, whole cells, or their molecules to solve problems or to make useful products. When coming to the agricultural biotechnology, uh, the large increase in the size of population has led to an increase the demand for resources and basic requirements such as food, shelter, clothing, etc. Another impact of increasing in population is the exploitation of the land for crops production. Thus, cultivation has been limited to a small area. In order to meet the demands with limited resources, we need to apply a great effort in biotechnology. In agriculture has changed the face of this condition. So the biotechnology is the use of biotechnology to modify or manipulate any biological system or living system for the development or improvement of products for various purposes. It is widely employed in different fields and agriculture, different fields. Agriculture is one of the among them. Along with agriculture, we also have the similar introduction for aquatic biotechnology also. So, aquatic biotechnology we will deal with after this agriculture biotechnology. We will discuss about aquatic biotechnology also. So that it is widely employed in different fields like agriculture. This biotechnology is also biotechnology is applied in different fields like agriculture and also aquatic biotechnology. And genetically engineered crop-based agriculture is an option. Others being agrochemical-based agriculture and organic agriculture. The revolution, the Green Revolution, was an initiation for increasing food production, but it could meet the growing demands. Later, the idea of crop variety improvement program was put forward. However, the agrochemicals seem to be unfeasible for farmers. In addition, the environmental issues related to them also reduced their use. So, genetically modified crops, GMO. Mm. Before going to the GMO, here tools of biotechnology. Here we have bioprocessing technology, and our next topic is also microbial biotechnology. So today, if time permits us, we will discuss about agriculture biotechnology, aquatic biotechnology, along with this bioprocessing, this microbial biotechnology. So, bioprocessing technology using whole living organism or components of them to manufacture desired products. Most common whole cells used are yeast and bacteria, mostly one-celled organisms. Most common components are enzymes, proteins that catalyze chemical reactions, microbial fermentation, and the cells isolated from animals and plants are also used to produce desired products. When coming to the genetically modified things, genetic engineering plays the major role here, that which is a technique of removing and modifying or adding genes to the DNA molecule to change the information which it contains. Is also specifically a recombinant it is also specifically known as a recombinant DNA technology, is in turn called as rDNA technology. The product of rDNA technology is known as a genetically modified organism, is also known as GMO. For example, gene for human insulin inserted into E. coli, a bacteria make human insulin a pharmaceutical, a biopharmaceutical product. And next, first, I want to give some uh, glimpses about genetically modified crops. Genetically modified crops, GMO, are the latest advancement in the agriculture field. These crops results from the alteration in the genetic makeup of the crops. This modification leads to a number of advantages in the crops. That includes there is a less loss after harvest, and the crops can be modified to having the additional nutrients value for human welfare, and these crops are modified to be highly efficient, the high yield with less usage of minerals, the decrease in the use of insecticides and pesticides, which led to which led to pollution in the environment. So, genetically modified organisms can decrease the use of insecticides and pesticides. That in turn impacts on the environment after reducing the pollution. A more tolerance in the natural stress like calamities and extreme temperatures, and also weather conditions, lack of water and minerals. One of the most common examples here we can see is a BT cotton. A BT stands for Bacillus thuringiensis, which, when introduced into the plants, develop resistant against the pests like bollworm and corn borer. Thus, genetically modified crops help in optimizing the complete process of agriculture advancement in biotechnology in the agriculture results in a variety of GMO, which includes the pest resistant plants, disease resistant plants, etc. Um, and also, it is nothing but a conventional plant breeding method uh, has been used to develop new varieties of crops for hundreds of years. However, conventional plant breeding can also no longer sustain the global demand with the increased population, decline in agriculture resources such as land and water, and the apparent plating of the yield curve of improvement technologies should be developed and utilized so that mutational breeding also used, and here in the agricultural applications, uh, improved crops like here you can see the corn, improve crops yields by introducing the genes that confer resistance to insects, a tolerance to herbicides, and resistant to environmental stress, drought, heat, and cold, and create disease and insect resistant trees to help meet demand for the wood products. And agricultural applications improve animal health, a better detection of disease, a more nutritious feed, a new and more effective vaccines, improve treatment for diseases. A medical and healthcare applications, new diagnostic tests like new generation of home pregnancy strip tests. And the other things we can discuss here is a chemical and environmental applications. Apart that, we also heard about the tissue culture techniques. It is also a part of agricultural biotechnology. A tissue culture is the cultivation of plant cells, tissues, and organs on specifically formulated nutrient media under the right conditions, an entire plant can be regenerated from a single cell. The plant tissue culture is a technique that has been around for more than 30 years. There are several types of tissue culture depending on the part of the uh, plant we selected, that is the explant we used. The first one is um, actually plants usually reproduce through the sexual means. They have the flowers and the seeds to create the next generation, like egg cells in the flowers are fertilized by pollen grains, pollen from the stamen, that is from the male part of the flower of the same plants are called self-pollination, or another plant called cross-pollination. Each of these sexual cells are containing the genetic material in the form of DNA. During sexual reproduction, DNA from both parents is combined, creating the offsprings similar to the parents in self-pollinated crops. In new and unpredictable ways, creating unique organisms in cross-pollinated crops. Some plants and trees, on the other hand, need several years um, before they flower and set the seeds, making plant improvement difficult. Plant scientists have developed the science and art of tissue culture to assist breeders in this task. So we have several types of tissue culture techniques. As just I said, depending upon the part of the explant we selected, um, that is the anther culture. Anther culture is a tissue culture method used to develop improved varieties in a short term, and pollen grain with an anther contains half dose of genome haploid, which spontaneously double, becomes like diploid, and during culture in some species, however, colchicine treatment is necessary to induce doubling. Uh, doubling. A doubling of the genome will allow the expression of recessive traits which were suppressed, emasculated, means masked or undetected in the routine plant breeding, so that anthers are placed in a special medium, and immature pollen grains, a pollen within the anther, divide and produce a mass of dividing cells termed as callus. Healthy callus are picked and placed in another medium to produce shoots and roots, are called the regeneration step. A stable plantlets are allowed to grow and mature in the greenhouse. A plant breeders can then select the desired plants from among the regenerated plants. Anther culture, F1 plants, first generation plants, which are progenies in a specific breeding objective, would allow many more different types of uh, regenerants. This is because the genetic constitution of the pollen will be more varied than those from the inbreeds. Thus, breeds will have a wider range of traits to choose from This technology has been employed in the successful development of doubled haploid lines of rice, wheat, sorghum, barley, and other field crops. Rice varieties developed through anther culture were released by the National Seed Industry Council in the Philippines in the year of 1995. The first anther culture, a derived salt tolerant variety, yeah, that is a PSBR C50 bicol, was developed by IRRI and released in 1995. The Philippine Rice Research Institute developed 8 salt tolerant varieties and 2 rain fed varieties. When coming to the micropropagation, is a tissue culture method developed for the production of disease free high quality planting material and for rapid production of many uniform plants, actively dividing young cells. Actively dividing young cell is also known as meristematic tissue or meristem, are placed in a specialized medium and treated with plant hormones to produce many similar sister plantlets. Since the meristem divides faster than disease causing virus, clean materials are propagated and hundreds of uniform plantlets are produced in short term and in short time. Through micropropagation, it is now possible to provide clean and uniform planting materials in plantations like oil palm, plantain, pine, banana, abaca, date, rubber, tree, field crops like eggplant, zuzuba, pineapple, tomato, root crops like cassava, yam, sweet, a potato, and many ornamental plants such as orchids, anthuriums, and micropropagated plants were found to establish more quickly, grow more vigorously, and taller, have a shorter and more uniform production cycle, the produce higher yields than conventional propagals. Here I want to discuss about embryo rescue also. Embryo rescue, the culture of immature embryos of plants in a special medium to prevent abortion of young embryos and to support its germination. This is used routinely in breeding parental lines having different or incompatible genome, such as introducing important traits of wild relatives into cultivated crops. The development of a new rice plant type for West America, that Nerica variety, new rice for Africa, West Africa, was resulted of wide crosses between the Asian Oryza sativa and the African rice Oryza glaberrima. It employs embryo rescue in the initial breeding and in the successive back crossing work followed by anther culture to stabilize the breeding lines. The new plants had combined yield traits of the sativa parent with local adaptation traits for glaberrima. Wild rices are a rich source. Wild rices are which rich source. Wild rices are a rich source of traits for resistant to pests and antibiotic stress. At the International Rice Research Institute, IRRI, embryo rescue is utilized and facilitated the transfer of bacterial blight resistance genes from wild rice, Oryza longistaminata, to variety IR24, resulting to a bacterial blight resistant line, that is IR BB21. Oryza rufipogon is a source of tungro resistant to number of rice varieties. At IRRI, a new super salt resistant rice was developed by saving the embryo produced in the cross between highly salt tolerated wild rice, that is Oryza uh, crocarata, with cultivated rice variety, that is IR 56. The research team, which is led by the scientist Dr. Krishrad Jena, has been attempting to cross the two rices since the mild 1990s and has only been successful. A fairly recently selected salt tolerant lines will be tested further by the farmers in salt affected locations for possible release within 4 to 5 years. A plant tissue culture belonging to the lower end of the agricultural biotechnology ladder, but the plant's ability to generate a new plant is an important requisite in the development of improved crops through agriculture biotechnology. A plant tissue culture is a straightforward technique, and many developments and many developing countries have already mastered it. Its application only requires a sterile workplace, nursery, greenhouse, and trained manpower. But unfortunately, tissue culture is laborious and labor intensive, time consuming, and can be costlier one. So that we have another techniques, that is molecular breeding and marker assisted selection. It is also called as MAS. Um, the process of developing new crop varieties requires many steps and can make 10 to 25 years, depending upon the crop. And now, however, applications of agricultural biotechnology have considerably shortened the time it takes to bring them to the market. It currently takes 7 to 10 years for new crop varieties to be developed. One of the tools which make it as much as easier for a crop varieties to be developed. One of the tools which make it as easier and faster for the scientists to select plant traits is called as marker assisted selection. The different traits and the physical features of plants are encoded in the uh, plant genetic material. Plant genetic material, the DNA that occurs in pairs of chromosomes, means the strands of genetic material, one coming from each parent, the genes which control the plant's characteristics are specific segments of each chromosome. All of the plant's genes together make up its genome. Some traits like flower color may be controlled by only one gene. Other more complex characteristics, however, like crop yield or starch content, may be influenced by many genes. Traditionally, plant breeders have selected plants based upon their visible and like measurable characters called phenotypes, a phenotype. But this process can be difficult, slow, that can be influenced by the environment and also costly, not only in the development itself, but also for the economy as farmers suffer crop loss. So that as a shortcut, plant breeders now use molecular assisted, molecular marker assisted selection to help identify specific genes. Scientists use what are called molecular markers, which are short strings or the sequence of markers, sequence of nucleic acids which make up a segment of DNA. The markers are located near the DNA sequence of the desired gene. Since the marker and the genes are close together on the same chromosome, they tend to stay together as each generation of plant is produced. This is called genetic linkage. This linkage helps a scientist to predict whether a plant will have the desired gene. If the researchers can find the marker for the gene, it means the gene itself is presented. So as scientists learn where each of the markers occur on a chromosome and how close it is to specify or specific gene, they can create a map of markers and genes on specific chromosomes. This genetic linkage map shows the location of markers and genes and their distance from other known genes, a scientist can produce a detailed maps in only one generation of plant breeding. Previously, scientists produced very simple genetic maps using conventional techniques. It was observed a long ago that as generations of plants were crossed, somatic traits uh, consistently appeared together in the new generations, a genetic linkage. However, since researchers could concentrate on only a few traits in each genetic map, using very detailed genetic maps or better knowledge of the molecular structure of plants' DNA. Researchers can analyze a tiny bit of tissue from a newly germinated seedlings. They don't have to wait for a seedling to grow into a mature plant or to test for the presence of specific traits. Once the tissue is analyzed through the molecular techniques, scientists know whether that seedlings contain the appropriate gene. If it does not, they can quickly move on and concentrate analysis on another seedling, eventually working only within the plants which contain the specific traits. Currently, molecular assisted plant breeding um, an agricultural biotechnology tool is already a routine step in breeding of most crops where the gene and the markers for a specific traits are known. This technique is being used in efficient efficient integration of important genes into various crops, including bacterial blight resistant in rice, increased beta carotene content in rice and cassava and banana, and submerging emergence tolerance in rice. And molecular markers are also used in determining the genetic profile of a line or variety. A random primers are used to scan the genomic constitution of the plant through molecular methods. The information is fed to a computer program that will analyze the relatedness of one line to another. This information on genetic diversity of lines is utilized in selecting for extremely unrelated parents, useful for hybrid seed technology. The information will also provide the details on the percentage of the line and possible traits and the unique identity of the plant useful for germplasm collection database. And as just we discussed, genetic engineering and genetically modified crops. What are the applications here? Genetic engineering techniques are also are only used when all other techniques have genetic engineering all the techniques have been exhausted, and when the trait to be introduced is not presented in the germplasm of the plant crop, and the trait is very difficult to improve by conventional breeding methods, it will take a very long time to introduce and or improve such trait in the crop by conventional breeding methods. A modern breeding, plant breeding is a multidisciplinary and coordinated process where a large number of tools and elements, a large number of tools and elements um, which is used of conventional breeding techniques, that is bioinformatics, biochemistry, molecular genetics, and molecular biology and genetic engineering are utilized and integrated. And the development of transgenic crops, although there are many diverse and complex techniques involved in genetic engineering, its a basic principles are reasonably simple. It is, however, very important to know the biochemical and physiological mechanisms of action, regulation of gene expression, and safety of gene and gene product to be utilized. The process of genetic engineering requires the successful completion of uh, different steps. Like we need six steps, a step one, a DNA or RNA extraction, that is nucleic acid extraction, and gene cloning, second step, gene design and packing, third step, and a promoter and selectable marker genes and reporter genes and enhancers. And third one is the fourth one is transformation and a particle bombardment, grow in fact particle bombardment we use agrobacterium tunifation mediated transformation. And after the insertion, uh, detection of inserted genes can be checked. And the last one is backcross breeding. If it is really needed, we will go for backcross breeding because genetic transfer technique is a usually conducted one. So we'll check what are these six steps. Um, the first one, nucleic acid extraction, like RNA or DNA extraction. Nucleic acid extraction, either RNA or DNA, like a ribonucleic acid or a deoxyribonucleic acid, is the first step in the genetic engineering process. It is therefore important that the reliable methods are available for isolating these components from the cell. If any isolation or procedure, the initial step is disruption of the cell of the desired organism, which may be viral, bacterial, or plant cell, in order to extract the nucleic acid after a series of biochemical and chemical steps. The extracted nucleic acid can be precipitated to form thread-like pellets of DNA or RNA. And the second one is gene cloning. The second step involved in gene cloning, second step is gene cloning, which is involved in RNA technology. There are basically four stages in any gene cloning program, gene cloning experiment, generated of a generation of DNA fragments, and joining to the vector of propagation in a host cell, and selection of required sequence in DNA extraction. All DNA from the desired organism is extracted. This genomic DNA is treated with specific enzymes called restriction endonucleases, or restriction enzymes, not endonucleases, restriction enzymes. These restriction enzymes cutting the DNA into a smaller fragments, which define ends to allow it to be cloned into bacterial vectors. Copies of the vector will then harbor many different inserts of genome. These vectors are transformed into bacterial cells, and the thousands of copies are produced using the information related to a specific molecular marker sequences and the desired phenotype. The vector harboring the desired sequence is detected and selected and isolated, and clones are produced. Our restriction enzymes are again utilized to determine if the desired gene inserted was cloned completely or correctly. Completely and correctly. The next one is a gene design and packing. Once the gene of the interest has been cloned, it has to be linked to a pieces of DNA that will control its expression inside the plant cell. These pieces of DNA will switch on, uh, is called as promoter, and switch off is called as terminator. The expression of the gene inserted. A gene designing and packaging can be done by replacing an existing promoter within a, a new one. The incorporating a selectable marker gene and reporter gene, adding gene enhancer fragments, introns, and organelle localizing sequence, among others. When coming to the promoter switch on, promoters allow differential expression of genes. For example, some promoters cause the inserted genes to be expressed at all the time in all aspects of plant, whereas others allow expression only at certain stages of plant growth, in certain plant tissues, or in response to external environmental signals. The amount of the gene product to be expressed is also controlled by the promoter. Some promoters are weak, whereas others are strong. Controlling the gene expression is an advantage in developing genetically modified plants. Selectable marker genes are usually linked to the gene of interest to facilitate its detection. Once inside the plant tissues, this enables the selection of cells that have been successfully incorporated with the genes of interest, thus saving considerable expense and effort. Genetic engineering used antibiotic resistance and herbicide resistant marker gene to detect cells that contain the inserted genes, cells that survive the addition of a marker inserted marker agents to the growth medium indicate the presence of inserted gene. Although increase in antibiotic resistance in humans and animals is unlikely to occur using antibiotic resistance markers, genes coding for resistant to non-medically important antibiotics are preferred. In addition, alternative types of marker genes have been developed which are related to plant metabolism, such as phosphomannose isomerase, xylome isomerase, and um, other others. When coming to the reporter genes, reporter genes are closed into the vector in close proximity to the gene of interest to facilitate the identification of transformed cells as well as to determine the correct expression of the inserted gene. The reporter genes that have also been used, which includes the beta beta glucuronidase gene is also known as GUS-A gene, which acts on a particular substrate producing a blue product, hence making the transformed cells in blue. The green fluorescent protein, that is GFP, which allows a transformed cells to grow under a green light, and a luciferase gene that allows cells to glow in dark, among others. Enhancers, several genetic sequences can also be cloned in front of the promoter sequences. Those are enhancers. Or within the genetic sequence itself, introns or non-coding sequences to promote a gene expression. An example is the cloning of a cauliflower mosaic virus, a promoter enhancers in front of the plant promoter. Once the gene of interest is packed together with the promoter, the reporter and the marker gene, it is introduced into a bacterium to allow for the creation of many copies, that is multiple copies of the gene package. The DNA isolated from the bacterial clones can be used for a plant cell transformation using particle bombardment technique. If, however, the use of used for plant cell transformation using a particle preferred in the plant transformation, the whole uh, gene package should be cloned in between two border sequences. of binary vector plasmid. This will allow processing of the agrobacterium so that only the transfer DNA, that is tDNA, will be incorporated into the plant genome. Our fourth step is transformation. In the transformation, the most common method used to introduce the gene packaging into the plant cells in a process called a bacterial transformation or a gene insertion, include a biolistic transformation using the gene gun and agrobacterium mediated transformation. A particle bombardment is a mechanical method which introducing a desired gene, the desired genetic sequence is cloned into the plant DNA vector and introduced into the plant using the gene gun or particle gun. As in the common gun, the gene gun uses minute particles of tungsten or gold as a bullet. These particles are coated with DNA solution and fired to the plant cells through the force of the helium gas inside a vacuum filled chamber. The DNA and the tungsten gold particles get inserted the cell, and within 12 hours the inserted DNA gets inside the nucleus and integrated within the plant DNA. The tungsten or gold particles are sequestered to the vacuole and eliminated later. So the transformed cells are cultured in vitro and induced to form a small plants called as regeneration that express the inserted gene. Next, the agrobacterium tumefaciens mediated transformation. The sharing of DNA among living forms is well documented as a normal or natural phenomena. For thousands of years, our genes have moved from one organism to the another organism. For example, Agrobacterium tumefaciens, a soil bacterium known as nature's own genetic engineer, nature's own genetic engineer has the natural ability to genetically engineer plants. It causes crown gall disease in a wide range of broad-leaved plants such as apple, pear, peach, cherry, almond, raspberry, and roses. The disease gains its name from the large tumor-like swellings, galls, that typically occur at the crown of the plant just above the soil level. Basically, the bacterium transfers of its DNA to the plant, and this DNA integrates into the plant's genome, causing the production of tumors that is associated with the changes in plant metabolism. Molecular biologists have utilized this biological mechanism to improve crops. The genes that cause the galls are removed and placed with genes coding for desirable traits. A plant cells infected with the bacterium will not form galls but produce cells containing the desired gene, which when cultured in a specific medium will generate into plants and manifest the desired trait. Then what is the main goal in any transformation produce is to introduce the gene of interest into the nucleus of a cell without affecting the cell's ability to survive. If the introduced gene is functional and gene product is synthesized, then the plant is said to be transformed. Once the inserted gene is stable, inherited, and expressed in subsequent generations, then the plant is considered a transgenic. Then our next step is detection of inserted genes. A molecular detection methods have also been developed to determine the integrity of the transgene, means the introduced gene into the plant cell. Here we have the technique of polymerase chain reaction, or PCR, is a quick test to determine if the generated transgenic cells are plants containing the genes. It uses a set of primers like forward and backward primers whose nucleotide sequence are based on the sequence of inserted gene. The primers are single nucleotides are incubated with the single strand genomic DNA, and several cycles of DNA amplification is conducted in a PCR machine. Analysis of the PCR products in agarose gel will show if the plants are really transformed when DNA fragments equivalent in size with the inserted gene is present and amplified. And the other technique, a Southern blot analysis, which determines the integrity of the inserted gene, whether the gene is completed and not fragmented at the correct orientation and with one copy number. The DNA coding sequence is probe binding to the single stranded genomic DNA of transgenic plant, which is implanted on a nitrocellulose paper. Autoradiography will reveal the transgenic status of the plant. One copy of the transgene is desired for optimum expression. And the other technique is Northern blot analysis, which determines whether the transcript or the messenger RNA of introduced DNA is present and is correctly transcribed in the process to bind to the nitrocellulose membrane. Labeled DNA is used to bind to the mRNA and can be visualized through autoradiography. And the other technique is Western blot. Western blot or protein immunoblotting is an analytical technique used to detect whether the transgenic plants produce the specific protein product of the introduced gene. A protein samples are extracted from the transgenic plants, are processed into denatured proteins and transferred to a nitrocellulose membrane. The protein is then probed, detected using the antibodies specific to the target proteins. Our last step, step 6, back cross breeding. If it is really needed, we will do this back cross breeding. Genetic transformation is usually conducted in elite or commercial varieties which already possess the desired agronomic traits but lacks the important trait of the transgene. Thus, once successfully conducted, the genetically modified plant will be easily recommended for commercialization if it shows a stability in several generations and upon successfully passing and fulfilling varietal registration requirements. However, some plant transformation may have been performed in plant varieties which are amenable to genetic transformation but are not adapted nor important in the target country. They may also be a sterility problems in transgenic plants. In such cases, conventional plant breeding is performed where the transgenic plant becomes the pollen source in the breeding program and the elite lines or commercial varieties as the recurrent parent. A backcross breeding enables the combination of the desired traits of recurrent parents and the transgenic lines in the offsprings. The length of time in developing transgenic plant depending upon the gene, crop species, available resources and regulatory approval, it varies from 6 to 15 years before a new transgenic plant or hybrids is ready for commercial use. There are some transgenic, genetically modified crops or biotech crops from 1996 up to the present. According to the Global Status Report, uh, presents an up-to-date record of the number of countries planting GM crops, the hectareage planted, the benefits of derived from the biotech crops, a farmer accounts of plants, biotech crops, as well as future prospects and directions of the technology. So far, 27 transgenic crops which are planted commercially, those are alfalfa, Argentine canola, bean, carnation, chicory, cotton, creeping bent grass, eggplant, flax, maize, melon, papaya, petunia, plum, and polished canola, poplar, potato, rice, rose, soybean, squash, and sugar beet, sugar cane, sweet pepper, tobacco, tomato, and wheat. With genetic engineering, more than one trait can be incorporated into a plant are called stacked traits. These are currently corn, cotton, and soybean crops with both herbicide and insect tolerant traits. Transgenic crops with combined traits are also available commercially, such as herbicide tolerant and insecticide or resistant maize and cotton. Stacking different genes of for one trait makes the crop more durable to resist the pest, disease, and tolerate more herbicides. Another strategy to improve a sustainability of the technology is the use of the refuse. Technology developers have studied effective refuse systems for specific transformation events. These are discussed to farmers extensively for a proper implementation and are monitored regularly to observe any resistant insects or weed. Uh, New and future initiatives in crop genetic engineering to the date, a commercial genetically modified crops have delivered a benefits in crop production, but there are also a number of products in the pipeline which will make more direct contributions to food quality, a clean environment, a pharmaceutical production, and livestock feeds. For example, rice with higher levels of vitamin A and iron and beta carotene, an important micronutrient which is converted to vitamin A in the body, and a long life banana that ripen faster on the tree and can therefore be harvested earlier. A maize with improved feed value, a delayed ripening papaya, a papaya, a ring spot virus resistant papaya, tomatoes with high levels of flavonoids, which are powerful antioxidant. Flavonoids are which are a powerful antioxidant, antioxidants. A drought tolerant maize and wheat, a maize with improved phosphorus availability, arsenic tolerant plants, insect resistant eggplant, and rice, edible vaccines from fruit and vegetables, low lignin trees for paper making, among others. Here, some crazy questions like what happens to DNA when it is eaten? DNA is chemically identical regardless of its source and is mostly regarded during industrial processing and the digestive tract. A small fragments can be detected in certain body tissues such as leukocytes, liver, and spleen. The daily human intake of DNA in food is estimated at zero point 0.1 to 1 gram. Estimates of the total daily transgene DNA that intake can be calculated, which assuming 50% of the total diet is from genetically engineered foods and transgenes represent an estimated point 0.0005% of the total DNA in food as zero point 0.5 to 5 microgram per day. So in 2009 July, the European Food Safety Authority released statements on the fate of genes and proteins in food and feed after ingestion. A rapid degradation into short DNA or peptide fragments is observed in the gastrointestinal tract of the animals and humans. And to the date, a large number of experimental studies with livestock have shown the recombinant DNA fragments or protein derived from genetically modified plants have not been detected in tissues, fluids, or edible products of farm animals. So, is the safety of genetically engineered food assessed? Yes. Before genetically engineered foods and products made from genetically engineered crops are approved for use, they have undergone a safety testing by the companies or the institutions that developed them. Data were reviewed by government authorities, government regulatory agencies, and scientific reviewers based on internationally accepted protocols. Frequently, genetically engineered food are also tested by outside groups and the results published in peer-reviewed journals. The process is comparable to safety assessments for pharmaceutical drugs and biomarkers. Pharmaceutical companies provide safety data that are subsequently reviewed by the US Food and Drug Administration (FDA) scientists to, mm, to the date. All genetically engineered products in the marketplace have undergone full reviews by regulatory agents and regarding safety and a content relative to unmodified forms and submitting the safety data in the developers a best interest given the legal liabilities incurred should a problem with the food arise following market introductions. And uh, the next question will be raised by many research scholars when I used to attend many seminars and conferences that are there are changes in the nutritional content of genetically engineered foods. And also another question is, does the bt protein affect humans? First I want to answer this question like, and do genetically engineered foods cause food allergies? Uh, the first question I want to discuss here is, does the BT protein affect humans? BT proteins are naturally occurring insecticide produced by the soil bacterium, that is Bacillus thuringiensis, used to control a crop pest such as larvae of butterflies and moths, beetles, and mosquitoes. Since 1920s, the crystalline inactive insecticidal Bt proteins from bodies inside the bacterium and become active when they are eaten by the target insect larva and cleaved. The active peptides bind to a specialized receptors in the midgut of the insect, creating holes in the gut membrane that cause contents to leak and kill the larvae. The precision of a different Bt proteins for their targets Bt protein. Their targets so that it cannot uh, produce any risks. So, but the only thing we can expect from the genetic engineering in agriculture is the increased crop productivity, uh, the increased crop productivity and enhanced crop production and improvements in food processing and improved nutritional value, a better flavor and a fresher producer and environmental benefits and benefits for developing countries. Just now we discussed about some risk, and one more thing also I thought of discussing, that is health related issues like allergens and toxins. People with food allergens have an unusual immune reactions when they are exposed to a specific proteins called allergens in food. About 2% of people across all age groups have a food allergy of some sort. The majority of the foods do not cause um, any allergy of some sort. Uh, Food allergies, people usually react only to one or few allergens in one or two specific foods. A major safety concern raised with okay, with this in the